Welcome to the 5 o'clock session for today. So today's session will be on developing synthetic materials to control natural biological processes. We've seen several different presentations this morning as well as in the previous day where people are using natural-based products or they're using a matrigel to act as a scaffold for different types of tissue engineering projects or just getting organoids to grow in culture so they could do different types of in vitro assays. But at times it's difficult to grow and propagate cells in a matrigel type of environment or in some other type of natural product environment because there's lot-to-lot variability that is a challenge to control. And in those cases, it's sometimes beneficial to move to fully synthetic systems where you can completely control the composition of your material so you can have hopefully reproducible results. And so the two people that we have presenting today are experts in their respective fields on creating synthetic materials and synthetic hydrogel. So the first speaker that we have today is Dr. Jay Rudra, and our second speaker will be Dr. Sylvia Zustiak. So for Dr. Rudra, he received his bachelor's degree in India and then moved over to the States where he received a PhD from Louisiana Tech. From there, he postdoced at the University of Chicago, and I was a postdoc in the same lab as Dr. Rudra at the time. From there, he moved and became an assistant professor at the University of Texas Medical Branch in Galveston. After several years there and some hurricanes and stuff, I think he decided to leave the Texas barbecue and move over to St. Louis barbecue where there's far less hurricanes. So he became an assistant professor, now is associate professor at Wash U at the McKelvey School of Engineering. So, Dr. Rudra.
Thank you, everybody, for being here today. It's my first time at this conference, and thank you, Josh, for the invitation. So I've worked with peptides almost all my life, grad school and postdoc, and even as a faculty member. They're short, minimalistic sequences, and we love that we can engineer them. And like, the theme here is to be able to control material properties by, making a few tweaks so that you have, the desired outcome in your biomaterial or your hydrogel. So today's topic is actually going to be about liquid-liquid phase separation and how we can use this, natural phenomena to generate materials that can now deliver therapeutics or even, vaccines and antigens and the likes. So this phenomena of liquid-liquid phase separation is very ubiquitous and has gained quite a bit of momentum in the last few years. We all know about cells and their membrane, and membranes allow things to pass through semi-permeably one way or the other. Liquid-liquid phase separation happens when you dissolve a protein in water, and then there is an automatic separation of the solute phase into droplets, and there's a continuous liquid phase. So in essence, this is driven by salt, pH, ionic strength. Any of these things could be a factor. And now we are kind of realizing that many functions, pathophysiological functions, are driven by these phase-separating condensates, including any number of cell-driven interactions, reactions inside the cell, and a few disease states that are defined by such phase-separating peptides. Now, this here is mostly driven, the interactions are driven by electrostatics, van der Waals, hydrophobic interactions. And for the most part, a lot of stuff that's been studied is in the context of proteins, whole proteins, or domains of some proteins. So what I'm going to talk about today is short synthetic peptides that are able to phase separate and able to encapsulate cargo and able to deliver that cargo efficiently into cells. So what you see here on the screen here is simply showing you that there is a monomeric phase, there's a peptide that's floating in buffer. Instantaneously, in response to pH or ionic strength changes, you end up getting the peptides kind of come together to form sort of a discontinuous droplet in that given aqueous environment. So the topic today is talking about histidine-rich big proteins. Histidine, this is going to be a lot about amino acids today. Histidine is one of those amino acids that's kind of unique. It is positively charged at some pHs, and it's not so positively charged at other pHs. We almost consider it as a positively charged amino acid along with lysine and arginine. Squid beaks kind of have a weird architecture to them in the sense that there is a hard end where you would expect to find chitin, and there's a softer lip on the bottom where you'd expect to see a lot of proteinaceous material, but it doesn't happen that way. So there is these proteins that have been derived from squid beaks that have the propensity to undergo phase separation in acidic environments, and this has been done extensively in the context of C-terminal domains from these proteins. And in mining these proteins, we've kind of, literature has come to report that there is a few consensus sequences that can actually phase separate even as short as 20 amino acids. So, we all know about elastin-like polypeptides that tend to undergo, sort of transition states between temperatures. Similar to elastin, histidine-rich beak proteins tend to phase separate given the pH and the ionic strength. And the consensus sequence here is GHGXY, where X can be leucine, valine, or proline. So it is a general sequence, GHGXY, and X can be now replaced with leucine, valine, or proline, and you need a minimum of 4 such repeats. Now, the histidine first undergoes deprotonation and then ends up forming sort of a cation-pi interaction with the tyrosine, which drives these droplets to form. So here's the 6 sequences that we made. So we made GHGLY, GHGVY, and GHGPY. We also, as synthetic chemists, like to make things with D-amino acids. D-amino acids are not coded for by the natural systems, but they have a few advantages in the sense that they are non-degradable. So whatever therapeutic that you're going to be delivering is going to last longer, and then immune cells are not recognizing the amino acids, so really don't see that it is a delivered substance, so you can have some immune evasion built into your technique. So these 6 amino acids were made, and each one of them is an enantiomer of the other, so we have GHGLY, VY, and PY, and then the 3D versions of that. Now, what we have here is a simple schematic showing you that all you need to do is add PBS at pH 7.4 to these peptides as dry powders, and then they instantaneously phase separate into droplets. Here's the data. For the leucine droplets, you would expect to see, that the enantiomers really shouldn't behave the same because one is right-handed, the other is left-handed. You see that the lowercase l indicates the D amino acids, so the top 3 panels are showing you the L amino acids, and the bottom 3 panels are showing you D amino acids. As soon as you add PBS, instantaneously they phase separate into these peptide droplets, and if you look at the size distribution here, your leucine droplets are about a micron, your valine droplets are about 0.8 microns, and then your proline droplets are about 200 nanometers. So this is, by changing a single amino acid, you have this exquisite control over the size of these droplets, and you can now try to figure out what the application is and where do you want to deliver these droplets into. The kinetics of assembly are also shown down here. The insets are showing you the kinetics of assembly over 24 hours. And then the bottom panels, the larger panels, are showing you a 3-hour time point. Essentially, it's cut off from that 24-hour time point. And what we're trying to show you is the kinetics of assembly are also very different here. The leucine droplets really start forming instantaneously, indicated by the high absorbance value at time 0. And then you see a drop, whereas the valine droplets are actually co-servating very slowly, and now they're building droplets into these little co-servates that kind of tend to grow over time. And given their super small nature, and the proline is one amino acid that is sort of weird because the side chain goes back and binds to the backbone, so it's always causes a kink in the peptide chain, and it's expected that the proline droplets would be super small, and the kinetics of the assemblies are shown here, and they're very drastically different assembly properties here. We just next wanted to know if there is a secondary, so before that, I just wanted to let you know that the droplet size and the formation of these droplets is very salt and pH dependent. So the dark circles are the L amino acids, the open circles are D amino acids. Again, L and D are mirror images of each other. The thermodynamics and the physics does not change. So you would expect the salt concentration and the pH concentration that would be the same for them to form the droplets. And you'll notice on the x-axis for the salt, they do form really good droplets at 150 millimolar, which is your physiological salt concentration. And if you look at the pH curves on the bottom, the formation of these droplets is the best when the pH is 7.4. Any fluctuation in pH to either side kind of reduces the droplet formation. And you'll also notice that the salt concentration does play a role in some cases. And sometimes, the valine droplets are pretty resistant to 1 molar salt, and then they still form, these droplets in ionic strength. Solutions with high ionic strength. Now we wanted to know what the structure is. What is keeping these droplets together? Because when peptides fold, they tend to fold into helices or beta sheets or keep themselves as random coils. So running a secondary structure analysis using circular dichroism spectroscopy gave us an identical trace for all three. And we had to go back, and I'm very familiar with helices and beta sheets, but not this new structure. So this apparently, the peptides have 25% tyrosine. It's GHGXY. So you have about 20% tyrosines, and tyrosine side chains can come together to form what's called pi-stacked bonds. And this particular spectra corresponds to pi stacking of the tyrosines within these peptide droplets that's holding them together. We also wanted to know when something needs to be injected into, through a syringe, shear thinning is an ideal property to have, because if you break these droplets up under shear forces, we want them to reform on the other end of the syringe, which kind of makes them, very structurally, intact. And to this, we reached out to Dr. Zistiak, and we ran a simple rheological experiment in her lab, where we did apply some shear force to these droplets, and then we looked at whether the droplets have any sort of stiffness to them, and you'll notice that the y-axis is the viscosity of these droplets. The leucine droplets were the most viscous compared to valine, and the proline droplets were the least viscous. And again, going from 1 millimolar to 2 millimolar in a concentration-dependent fashion, the viscosity can be increased. And then if you apply a shear, this is a continuous shear rate, so which means that they can actually, you can see the curves going down with increasing shear rate, which kind of tells you that these are shear thinning properties. So you can really shear them into liquids, and if you let them go, they kind of get back to their original viscosity or stiffnesses. We have to do this over multiple cycles, but this is a good sign. When the viscosity goes down with shear rate, it means that they can fall apart, and then hopefully they can come back up when you give them a rest. Moving on, we just next wanted to understand what are these actual contacts that are happening? How are these droplets actually staying together in solution? And this is a computational simulation of 10 peptide monomers thrown into a box and simulated for 1 microsecond. And on the x-axis, you see the individual amino acids, and then the y-axis, you see the same individual amino acids. And the hotspots are where you expect the probability of most contact to happen. So those residues would be, again, this is a predictive computational modeling. In here, you're looking at the probability of interaction. That is not a real interaction. So you're looking at the probability of which amino acids in the given matrix are responsible for holding these droplets together. And what you'll notice is that in the case of the leucine droplets, you see a few hotspots that are tyrosine and histidine rich. And then in the GV, you'll see a lot more hotspots that are interacting between glycines and histidines for some reason. And there's also some hotspots in the GP peptide when you replace the proline, that there's a few hotspots all the way at the C-terminal end. And then we also wanted to understand if the radius of gyration of these molecules is being maintained in solution. So if something is going to form a droplet, we expect to see that it actually condenses down. So if the radius of gyration for a monomer is, say, x, and then it comes into contact with its own kind, you'd expect the interactions to be actually crunching the radius of gyration for the individual molecules. But that's not what we saw. You'll notice that for all cases, the radius of gyration is higher in the droplet state than it's in the monomer state. So what that tells us, that the individual peptides were actually constricted when they were alone in the salt solution, but as soon as they started to form droplets, they sort of relaxed and they kind of opened up. So this data sort of is expected behavior for phase separating systems, and this is confirmation that, that these peptides do undergo liquid-liquid phase separation. Now, this is The next slide, looking at the encapsulation efficiency of these droplets. We wanted to know what can you put into these droplets and how can we deliver them. So we started with the simplest protein of all, GFP. And all we did was dissolve GFP in PBS and added it directly to the powder, or the peptide powders. And the encapsulation efficiencies were drastically different. The leucine peptide actually encapsulated 80% of the cargo, 70% for valine, and proline was very poor at encapsulating any molecules, partly because of, I think, the super small size and then the weirdness of proline that was built into the system. But you'll notice that the entire fluorescence is concentrated in the droplet and not in the background, suggesting that the GFP was trapped into these liquid-liquid phase-separated droplets rather than being free. So now this gives us an avenue. Now, can we now use these droplets for delivering not GFP, but something more interesting into a cell? Here's an idea that we kind of looked at, and one of the key components of liquid-liquid phase separation is that they tend to fuse together, and fusion is not always a good thing. The two droplets can come together and become one droplet. There's a time-lapse image shown on the surface, and I don't know why my video is not playing. Could you hit the video for me, please? And in that video, with the arrows where you're marked, in those white arrows, you can see two droplets actually coming together and fusing into one large droplet, and the time-lapse images are shown on the top. Now, fusion can be a good thing, it could be a bad thing. If you want to deliver cargo independently, you really don't want fusion to happen. And this fusion behavior was only visible in the valine droplets, but there was no fusion at all the fusion happened in the leucine droplets, but not in the valine droplets. So the question was, now can you deliver only one cargo, or can I put multiple cargoes into one single droplet? And I'm going to use the word condensate and droplet interchangeably, so please forgive me. So we picked 3 proteins labeled with fluorophores. One of them is mouse IL-4, which is a cytokine, which is a red. The green one was FITC, labeled with anti-mouse H2KFB antibody. And the third one was also a protein, cytokine, interferon gamma. So what we did was we manufactured leucine droplets with individual colors. So only one protein was trapped into these droplets, and the droplets were mixed together. And we just waited an hour to see what happens. Now, as the droplets fuse, you should start seeing secondary colors like magenta, cyan, and all these colors that come from mixing red, blue, and green. And you'll notice that you have all the 3 expected colors in there, and a complete merge image shows you almost white droplets, suggesting that the colors have now mixed. So which kind of tells you that the droplets are capable of encapsulating individual proteins or therapeutics, and when they fuse, they do so without leakage. And the second experiment was to mix all of them together, and now you should have a gray formulation. And in that sense, what you have is the same thing. Whether you mix them independently or you mixed all 3 therapeutics together and encapsulated them, there's absolutely no leakage out of these droplets, and you'll notice that the fluorescence is concentrated within the droplet, which kind of makes them exciting, for multiple cargo encapsulation and delivery simultaneously. Where do they go? Can they enter a cell? So here you're looking at a dendritic cell that has been treated with these droplets, and the cell membrane is stained with a pink dye coated for sugars on top. And then you'll notice that in the single plane image, and there's a 3D bottom view, that the droplets are inside the cells. The leucine droplets are on the left. The valine droplets are on the right. The droplets can enter cells effectively to deliver the cargo. And then on the bottom, there's a free peptide or the free protein control. And then we also looked at transfer sections, and we were kind of curious. How do they enter the cell? And then there was one instance where we caught a leucine droplet. I wish I had a laser pointer to point out to you, that kind of stretched out, and you can see it going through the cell membrane. It's the second droplet from the left, and it was stretched out, and as it was going through the membrane, and you can see these leucine droplets, they're not spherical anymore. They're kind of stretched out because they're embedding themselves in the membrane and traversing the membrane into the cell. And then we can actually show you a 3D image here, all the XYZ planes, and to the far right corner, you'll see this, the front view and the top view of that leucine droplet that's trying to get into the cell via the cell membrane. And this behavior was not observed in any other formulation, not the valine, not the proline, but the leucine droplets had this weird behavior where they simply fused to the cell membrane and they just entered the cell. Now, does this mean that the entry of these cells, these droplets into cells is energy independent, independent of ATP, temperature, or any of these other factors? So we went back and we quickly looked at this using flow cytometry, and the study was simple. You incubated cells with these droplets at 37 degrees like you would normally do, or at 4 degrees, or in the absence of ATP, or in the presence of a complete actin depolymerizer, latrunculin A. So the cell should be immobile, and if anything is happening, the droplets should be entering the cells in a completely energy-independent fashion. And the data does confirm that. The red bars are leucine, and you'll notice that the control, ATP depleted, latrunculin treated, and the temperature drop does not really affect the entry of the leucine droplets, but it does affect the entry of the valine droplets. And the lighter red and the darker, the lighter green bars are just the enantiomers, the D-amino acid versions of the same, showing that the entry is energy dependent in the case of valine droplets, but somehow the leucine droplets can enter these cells in an energy independent fashion. So this was very exciting, because this is sort of, gives us a leverage in trying to deliver therapeutics at low temperatures, or in cases where there is some ATP depletion. I can't think of a biological situation where that would be, but this is some exciting data. Then we wanted to understand, like the first experiment we did was, can we now deliver a real therapeutic, like a transfection agent? And for this purpose, we already showed that we could encapsulate the protein GFP. Now the question became, can we encapsulate GFP mRNA and a GFP plasmid and deliver it to HEX cells for transfection? And here's the data. The plasmid only is shown, and lipofectamine does a beautiful job transfecting, but the cells are dying and they're detaching. And the leucine droplets effectively delivered the plasmid into HEX cells, and you can see the protein expression at 48 hours, but the valine droplets were not effectively, transfecting these cells. So given that small change in one amino acid, you get these droplets with different behaviors, where one of them is energy dependent, the other was energy independent. One can transfect, but the other can't. But in the context of some other examples, I'll show you that the valine does a better job than the leucine. So this is an open field. The next set of data here showing is mRNA for eGFP encapsulated and delivered. Lipofectamine, again, does a great job, like you'd expected, but there were a few cells we found in the leucine-treated group that showed some good mRNA transfection. So these condensates can now be used as effective transfection agents, and they do not kill cells. But the efficiency is nowhere near as much as lipofectamine, but then that depends on your application and how much of a knockdown you actually want. The next question was, can we deliver antigens in the setting of a vaccine? And for this purpose, we choose chicken egg ovalbumin. It's a very ubiquitous model antigen used by immunologists because it's been well studied. The peptides that are the T cell epitopes for the CD8 and the CD4 T cells are well known. And we chose to do a different sort of an experiment here. We chose to use the ovalbumin protein modified by a BodyP dye. BodyP has this unique ability to only fluoresce in acidic conditions. So the fluorescence on top of the acidic conditions will only come if the protein is broken down into individual peptide fragments. So if you see fluorescence inside a cell, it means that the droplet was able to capture the ova, deliver it into the cell, and end up in the lysosomal compartments where things are broken down for presentation. So if you see green fluorescence, it means that the ova protein was delivered into the cell. And this is our data showing you the green, the red is lysotracker, the blue is hash staining for the nuclei, and you have L, V, and P vertically arranged, and you see green fluorescence in all three conditions, showing that the droplets were able to encapsulate the antigen, deliver it into the cell, and end up in the endolysosomal compartments for actual processing and presentation now to T cells in the setting of a vaccine. Now, I just wanted to give you a small primer on how do we do these readouts and how effective is a vaccine. This is what we call an in vitro vaccination assay without going into an animal. And I just want to quickly talk about the pathway. So any protein that is entering into the cell from the outside goes to the MHC class II pathway, ends up in an autophagosome that fuses with the lysosome, and then the protein gets broken down, and peptides get loaded onto the MHC molecule and presented to the surface of the dendritic cell, which now a T cell can look at and then start doing it. So how do we measure this in vitro? If we have a simple assay, what we call an in vitro antigen presentation assay, we take dendritic cells, treat them with the antigen-loaded microdroplets or protein peptide droplets, and then we just overlay the dendritic cells with the cognate T cells. And in this case, we are overlaying T cells that only recognize one peptide from this whole protein. And that interaction will produce IL-2, and the IL-2 is now read out in a simple ELISA, and the amount of antigen presented on the surface corresponds to the amount of IL-2 produced. So higher the amount of IL-2 produced, more the antigen presentation, which is a good thing as far as vaccines go. And to be able to do this experiment, then we went back and we did the same experiment where we treated the cells with these droplets loaded with ova, and then we overlaid the dendritic cells with T cells after 24 hours, and then went back and measured the levels of IL-2. And now in this data, you'll notice that valine outperforms leucine. So the valine droplets were a lot more effective in producing higher amounts of IL-2 and in a time-dependent fashion. And this is what I was telling you about the purpose of using D-amino acids. The dark green bars are valine, natural L-valine, and the shaded green bars are the non-natural D-valine. Now, since D-valine droplets cannot be degraded by proteases, they persist for longer times, which means a slower release of antigen, which means more IL-2. So that's sort of an example of where you can leverage these D-amino acids for delivery and prolonging the lifetime of the therapeutic that goes in. And you'll notice that this is a continuous time treatment for about 72 hours. So this is, once we treat the cells, we wash them and then overlay, so there's no continuous antigen being presented. So what entered the cell in that 24 hours of treatment persisted for either 24, 48, or 72 hours. So this was a very, first time trying to understand how these droplets can be used to be able to engineer some vaccines down the road in the context of infectious diseases or cancers. That being said, just also wanted to let you know that we did some control experiments to show that this effect is real. Now, you can block MHC antigen presentation by multiple ways. The first thing you do is you stop lysosomal acidification. If the lysosomes don't acidify, the protein doesn't get broken down. If the protein is not broken down, there's no presentation to T cells. No presentation to T cells, no IL-2. So it's a simple concept, and we use 3 different inhibitors to show that this effect is real. And in the presence of the inhibitors, the intracellular fluorescence goes down, because remember, ova only fluoresces when it's broken down. So no protein breakdown, no fluorescence. And we show convincingly that in the presence of inhibitors of lysosomal acidification, we use 3 different inhibitors and showed that the antigen processing and presentation is diminished, presentation to T cells is diminished, and then therefore not much IL-2 coming out of the systems compared to normalized untreated controls. So overall, the kind of very exciting area to be in, and the next work in progress is that we're now using ChatGPT to teach it how to mine these sequences for us. 18,000 proteins taken, and we told them, okay, we know that some form LLPS, some proteins don't form liquid-liquid phase separation droplets. These are the ones that form. These are the ones that don't form. Now search for sequences that will help us figure out if a short peptide can form a droplet or not. So here's our outcome. We tried about 4 different sequences, and luckily 3 of them did form liquid-liquid phase separation droplets, and it's very excited to see what the molecular properties are, what can they be used for, how do we exploit them to put in therapeutics and cargoes, and drive some cool applications down the road. I'd like to thank my entire lab, my collaborators, particularly on this particular project. It was driven by my 2 postdocs, Ushashi and Arban, and collaborators on this project, Dr. Heather Struckman for the confocal, Dr. Zastiak's lab here for the rheological properties, and then Dr. Mondel for some computational analysis of the proteins for us. And with that, I'll take any questions.
Got time for a question or two?
Have you tried antifreeze peptides?
No, no, I have not. I don't know what they are, and I'm happy to chat with you, today.
For freezing cells, but you don't want the cells taking it up. So antifreeze peptides, now they are like smaller molecules.
No, I've
No,
not.
yeah.
I've not done that.
Other questions?
How big can the size of your cargo be?
So we put in proteins. We put in three big proteins. So I would say at least 500 kilodaltons was in one droplet. Yeah. So yeah, they're big. They're a micron long. They're a micron in diameter, so they're pretty big droplets.
Yeah, really nice work. So I'm trying to get better understanding on the uptake. So you kind of showed with the leucine interact. Now, did you say it's fusing with the membrane, or it's?
There's a couple of events we saw where it fused, and there's a couple of events that we caught where it just stretched itself out through the membrane and went to the other side.
Okay, because the issue is going to be processing, right?
Yes.
So depending on how it enters the cell,
Yeah.
it's going to change the processing.
So now I've shown you two sets of data. The leucines droplets transfect great. So my understanding is that they probably fall apart in the cytoplasm where you want the transfection, components to be released. The valine droplets really don't fall apart at all, and they don't fuse. They have no fusion
Yeah.
capacity. Even individual colored leucine valine particles don't fuse. The droplets don't fuse. And somehow they go all the way to the lysosome, and you get a better immune response or reading from them. So again, hypothesis at this stage is that if you want something delivered cytoplasmic, go with the leucine droplets. You want something in the endolysosomes, go with the valine
with the
droplets. That's
valine.
what my
Okay.
data is telling me for now, but I can't speak,
Yeah,
with full
and the
confidence.
term great is always relative, right?
It's
So-
always relative.
So that was the other thing too. You were comparing with lipofectamine and then the droplets, and you made a statement that like the lipofectamine, you're getting all the cell death,
Yeah.
which is a little bit of an exaggeration. But
For 48
but
hours-
you were saying that with the droplets, they're all alive. So
They're 100% alive.
100% alive.
100% incubated for days at a
So I
time.
would say with your plasma delivery, your efficiency is not very good. If
It's
you have
not.
100% viability.
Nope, it's not nowhere near lipofectamine at all.
Okay.
I
So
we
that's
acknowledge
kind of
that.
what, apples to oranges and
Absolutely.
things like that.
Agreed.
And, so, can I? So, why not? So, the other thing is, how do you envision it? So you're talking about vaccines and immune. So the critical aspect with any immunotherapy vaccines is going to be how much you got in there, right? There's a balance of overstimulation versus pro-inflammatory response. So how do you envision doing this? Because all your testing, and I understand you need to do this, is in a perfect world, right? You're controlling your cells, you're controlling your droplets, you're adding them directly to the target. When you go into actually using this, you have so many different things that are going to interfere with that.
Yes.
And so have you thought about that at all? Or?
Yes, and it's not different from the lipid nanoparticle vaccines we all got 4 years ago. Once those lipid nanoparticles go in with spike protein, who knows where they go? So
This is the problem.
It's a problem, I understand. If you wanted to decorate the surface of the droplets with the targeting moiety, if you know the ligand specific for an immune cell receptor, definitely doable.
Right.
But I'm not sure whether this platform is the right one. These are phase-separated, barely hanging on together droplets. These are not nanoparticles. These are not polymeric nanoparticles that are more rigid. So these are...
This is where it becomes an issue with vaccines because the question is whether you have an antibody response or a cellular response.
Yeah.
You were trying to measure a cellular response.
That is the goal
Which-
because there's enough antibody triggering systems out there, not many T-cell sort of systems.
Maybe we should talk later.
Yeah, sure. Thank you. Thank you
All right.
very much for
Thank
this
you, Dr. Rudra.
opportunity. Yeah.
To keep things moving. So up next, we have Dr. Sylvia Zustiak. She received her bachelor's degree and a master's degree in Bulgaria, and then came over to the U.S., did a PhD at the University of Maryland, Baltimore County. Decided to stay kind of in the greater Baltimore, D.C. area, went a little bit west, did a postdoc at NIH, and then decided to go much farther west to St. Louis. So she's been at St. Louis University since 2013. She started off as an assistant professor there in the Department of Biomedical Engineering, and she is now a full professor, along with being the associate chair of the Biomedical Engineering Department. On top of that, she's a co-director of the SLU Institute for Drug and Biotherapeutic Innovation.
Thank you. So I have my own timer because I know I'm between you and dinner. All right, so today I'll talk to you about synthetic materials, but more about hydrogel materials, not peptides. So first of all, my lab works in the drug discovery space, and we have two distinct umbrellas of projects. One of them is drug discovery, one of them is drug delivery. So if you don't know enough about the drug discovery funnel, as we call it, what happens is you start with hundreds of thousands of molecules, and then you end up with one or two. 15 to 20 years later, hundreds of millions of dollars, and just like a very laborious process. What I wanted to point out to you as an in vitro biology conference is that, we go from cell culture on 2D to animal models. One is simplistic, one is too complex, ethical concerns, and so forth. So as in vitro biology, you probably appreciate that we can make in vitro models. And if we put those in materials, those now are more physiologically relevant. You can use human tissue, you can use human cells, and they can replicate the in vivo environment more truthfully. So what will happen in that case is now that if you have a better model, you can screen your therapeutics, your potential therapeutics, and you can potentially avoid false positives, false negatives, and you can address the problem of basically failures during clinical trials, which happens due to lack of efficacy or unexpected side effects. So the other part of my lab then focuses on the drug delivery, and that kind of goes more into the lead optimization space, where you're trying to improve the PK properties of the drug. And I'll talk more about that, but we focus on localized delivery devices, and those are hydrogels, which are injectable. Before I move any further, all I'm going to talk about today is one material. We work in the lab, but I'm going to talk about one of them today, which is our workhorse PEG, or polyethylene glycol. So all of you, if you haven't heard about PEG, it's just simply because you probably weren't looking at the ingredients on the drugs that you are taking, or maybe food that you are eating. It's all around us. It's in cosmetics, in food, and so forth. It's very biocompatible. But there is so many different formulations of PEG. So what we work in our lab is something that gels by something which we call microtype addition. And so basically, you have a multi-arm polymer that has a double bond, like an acrylate or vinyl sulfone. You mix this with something that has a thiol, also a PEG thiol, or something that has a cysteine if it's a peptide. You simply mix this up and down together, and it forms a gel. Voila. And when you do this, you can put your cells or your biomolecules of interest inside that hydrogel precursor solution, and the hydrogel forms around it. It's a very physiological and also highly selective chemistry, so we can put anything in it, basically. We make our own cross-linkers, but I won't talk about this today. So we use this first as injectable and bioresorbable drug delivery devices. What that means, we have different formulations. In one formulation, for example, you can take that liquid formulation, you can inject it, and in situ, it will solidify. And when it solidifies, it's going to trap those proteins inside. You can inject it anywhere where you can basically put a needle through. Typically, subcutaneous is a very good site of administration. And then over time, what you'll have is you'll have your protein, let's say, therapeutic, delivering sustainably from that device. And also, delivery happens basically because of degradation or diffusion. We have a different injectable formulation, which is microspheres. And now in our lab, I think we specialize in making droplets. We have many different ways in which we can make them. I'm showing you here a microfluidic device, but we have electrospraying and a variety of others I can talk to you about later. But what those allows us to do, we can make microspheres, which we call microgels or droplets of various sizes and polydispersity. And those are small enough that they can be injected through a needle. Also, our PEG gels are shear thinning, and they're also very pliable, like squishable, so that they can be injected without much force. And I will show you some applications of that. And the last thing that we do, so what I just showed you earlier is just our PEG hydrogel. So if you encapsulate, it's a nanoporous structure so that if you put any kind of a large molecule, like it's a protein inside, it will be released from the hydrogel just to pure diffusion or degradation. But that's not enough sometimes because you might have burst release. And so in this case, we have also designed those nanocomposite materials. One example that I'll show you today is we're using these called nanosilicates. So if you can imagine, those are like little coins. And those coins have charges. The faces are negatively charged, and the edges are positively charged. So what that does is now if you can put a protein, and the protein, they typically have charges, they can adsorb onto the surface. You can complex those, as shown in the steps that you're seeing. And so once you make the complex of those nanosilicates with your protein, you can encapsulate those inside the hydrogel. And now you have another lever to control release, not just diffusion and degradation, but now you can also use electrostatic interactions or adsorption and desorption. So we use also this a lot in the lab. I'll just give an example with 3 model proteins, which all of you probably have seen or heard about. One is serum albumin, ubiquitous in blood. This is bovine serum albumin that we are using, ribonuclease A or RNase, and then lysozyme. Why we chose those? Because bovine serum albumin is negatively charged, and then the other 2 are positively charged, but they have different charges, as you can see in the table. They also have different molecular weights. And if you look at the release profiles, now you see the dotted line and the solid line. So if you look at the dotted line, these are just the proteins being released from the plain PEG hydrogel. What you're going to notice is something very typical for release from hydrogels. You have large burst release. So about 50% of the protein is going to be released within the first maybe couple of hours, and then the rest you're going to have your sustained release. It's not bad for every application, but sometimes it's a negative. So if you now have your nanosilicates inside, now you have electrostatics. And so you can see, for example, if you pay attention to the green line, you can see that a very small protein such as lysozyme, but very highly charged, now is released extremely slow because now the release is controlled by electrostatics, by desorption. You can see this just a little bit here more like visually. First, if you simply look at the schematic at the bottom, you can see that your coin-shaped nanosilicates. And if you have a positive charged protein, for example, like ribonuclease A, that's going to be adsorbing onto the faces, the negatively charged faces of the coin. Then if you have something which is positively charged, with negatively charged, I mean as a BSA, it's going to be adsorbing onto the edges, which is a lot lower surface area overall. And we did some other studies with fluorescence correlation spectroscopy. I'm not going to go into this, but basically what we also saw then is that the diffusion and bound fraction can also be controlled by the concentration of the nanosilicates. And again, you can see that for a protein which does not adsorb as tightly, you have less fraction bound and faster diffusion than something which adsorbs more tightly, such as lysozyme. I promise I'll switch topics soon, so you'll wake up, but I have just a couple more slides on the release. And so this is just the conclusion for this one little thing. It's just to show you that we can make any kind of designer materials, right? We have our hydrogels, we can put nanosilicates inside, and that just gives us another lever to make these plug-and-play devices so that you can control release for any application. And by the way, I did not mention it because I took the slide out, but our materials are also degradable, as I mentioned, and they can degrade from about 12 hours to 32 days or even longer, depends on the composition. So again, we just have a huge library of materials that we can use for a variety of applications. One application I will talk about today, and if I have time, I'll talk about a second one. But the first one is a mouthful, so just bear with me. So these are just, again, injectable polyethylene glycol microgels. So we call hydrogel microspheres microgels for short. Those are also super lubricious, loaded with platelet-rich plasma to treat knee osteoarthritis. A mouthful, right? So if you have heard about platelet-rich plasma, that's something that's currently used in the clinic. It's really useful because it's autologous, so the physician can simply take blood from you, spin it down, take the plasma, take the buffy coat, lyse the platelets, and voila. Now you have this concoction that has 300 plus molecules, cytokines, growth factors, lipids, which supposedly are good for you, good for tissue regeneration. However, its PRP is great. It has been used in a variety of applications, actually wound healing, not just osteoarthritis, cosmetics, and so forth. But the problem, like for us, for the knee osteoarthritis in particular, is that the synovial fluid in the knee exchanges every couple of hours. So currently, if you can imagine, you inject something intraarticularly, 2 hours, it's gone. Your cartilage is very negatively charged, so very hard for anything to penetrate through the cartilage. So what we're basically proposing is, well, you simply want to increase the residence time of PRP inside the knee. You have to include it into some kind of a delivery device, in this case, injectable microgels. It's going to stay there longer. So hopefully, it's going to have a beneficial effect. We also make our materials super lubricious because, as you probably know, synovial fluid in the knee, once you get osteoarthritis, becomes very liquidy. So it doesn't have enough lubricity, so we also want to make this lubricious. And by the way, all of us, like if we're lucky to live over, to be 85 or more, we'll have some form of osteoarthritis. So it's really, really relevant. It's a progressive disorder that currently has no treatment, even though we have been working on this for many, many years. Actually, many. More than I can count. So PRP in this case, just a little bit of background. We looked at different formulations of PRP that I'm not going to go into today, but the bottom line is that we can release our PRP from the microgels in a sustained manner. We also did some multiplex analysis to look at individual proteins, like let's say EGFP and PDGF, and we saw that even though we did have the burst release as expected from previous data, we still had enough protein left to be released even at the degradation time, which in this case was 11 days for this formulation. So we can release PRP. Well, okay, what can it do? We looked at patient-derived chondrocytes. To make them osteoarthritic, we also treated them with TGF-beta. And so then we exposed them to PRP. What you see here in the dark red color is a bolus PRP, and then we also have our released fractions. Day 1 simply means that we are taking all of these bolus released molecules. Day 4 is anything between day 1 and day 4, so everything after the bolus release. And then day 11 is anything towards the end until complete degradation. So when we looked at the fold change, which means chondrocyte proliferation, we saw that one particular fraction, which is a day 4 fraction, really increased proliferation, and the other ones didn't have much of an effect. But then, of course, I did many other studies, and 2 of them, which I'm showing here today, are the expression of MMP13 and NF-kappa B. So MMP13, this is simply a matrix degradation. And so we saw that all of our fractions plus the bolus PRP were able to decrease matrix degradation, to suppress matrix degradation gene expression, and then also suppress NF-kappa B, which means suppress pro-inflammatory cytokines. So basically, I'll talk about this in a second. So the next thing that I wanted to mention is that our microgels are also super lubricious. And so how did we do this? With help of some of our collaborators from chemistry, we were able to synthesize a super lubricious polymer coating, which has 2 components, DMA and SBMA. So DMA is kind of like the muscle glue, and so that is responsible for absorbing onto our PEG microgels. And then the SBMA is a Zwitter ion, which is basically very, very well hydrated molecule, which gives it super lubricious properties. So we simply put our microgels in, deep coat it in the polymer, and they become lubricious. You see a little bit of fluorescence picture. So we also made the copolymer fluorescent by rhodamine just to show that we have a nice efficient coating on all of our microgels. And when we did a little bit of testing to show the lubricity. And by the way, I'm showing you a custom tribology setup. So once I started to do tribology, we realized that we don't have a tribology, first of all, set up in St. Louis. But second of all, even a commercial one, you need huge samples, huge volumes of samples, and we can't do this with biological samples. They're extremely expensive, so we designed our own. We custom retrofitted our rheology, our rheometer, with a 3 ball-on-a-ball setup, so that basically when you spin the upper plate, you can get coefficient of friction. Like, I'm not going to do the math, but papers hopefully soon coming out. So when we did this, we compared now just regular buffer to our uncoated and coated microgels, and also synovial fluid from patients, because we also have that. And we saw that our coated microgels were actually more lubricious or lower coefficient of friction than even synovial fluid. So yay, great, right? So we also saw that as we increase the concentration of that copolymer, we can make them even more lubricious. So that was exciting. And I won't go any further into that study, it's ongoing. But just very quickly, what I showed you so far, at least, is that PRP can maybe, right, be clinically relevant. Like we so far have seen this, it's just mixed results. But also that we can have kind of these dual action gels, not just deliver PRP, but also make the synovial fluid lubricious again, which is kind of a big problem actually for patients, actually which causes the pain for patients and eventually leads to total knee replacement. Okay. I'm keeping here my time, so I will just fly through these next 2 slides. This was another example of a drug delivery with a different collaborator at SLU, and this was for a childhood disorder for an enzyme replacement therapy, more QA. I won't go into this, but the whole thing here was that we are trying to deliver an enzyme, which is lysosomal storage. This is like, so it's a lysosomal enzyme, which also means that that enzyme is only stable in the lysosomal environment. So imagine trying to deliver something which is only stable at pH 5, and you have to have it in physiologically relevant environment. We did a lot of studies, which I'm not going to show today. We have published papers on that. But basically, we were able to design a hydrogel formulation that improved the stability of that enzyme, which otherwise in a solution within 24 hours is completely loses activity. In our hydrogel, it was able to be stable for up to 7 days. We actually also even up to 28 days, even though at the end it was like 7% activity. But it was able to actually be stable for a very long time, and we were able to achieve sustained release. We showed this in mice as well. You could inject the gel subcutaneously, the gel stays there. And if you look at the mice, you have that fluorescently labeled GALS enzyme stayed in the mouse for a very long time in the gel formulation, but it completely disappeared when gel was not present. So that was like another quick story. Again, no background on that disease. It's no time. Today, some of you saw a presentation from my students, so I probably won't spend much time on that as well. But this is completely switching gears now. Completely. Forget about drug delivery. This is now like drug screening platforms. So, right, like we use the same material we used to encapsulate proteins and deliver proteins. We can use similar formulation, but now we can encapsulate cells. And now you can use these cell scaffolds or in vitro models to test therapeutics on. This is what it's about. So this was about specifically we work on glioblastoma for a variety of reasons, which my student talked about today, but this is like a really deadly brain cancer, no treatment, chemotherapy does not work. What's more interesting for us, and for me in particular, is from the materials perspective. The GBM microenvironment is extremely complex, like every other tumor. What's interesting for those cells, though, is that when they form a secondary tumor, which is actually what kills the patients, not the primary tumor. The primary tumor can be resected. But the secondary one kills the patients. When the cells are migrating away from the original tumor, they don't just go through gray matter. They follow white matter tracks. They follow the perivascular niche. In fact, if you put something stiff in the brain, they'll follow that. They do seem to follow stiffness interfaces. And so my lab has been trying for a very long time to design a model that has such stiffness interfaces so that we can observe the cells. I will fly a little bit through this. This is one model that we made. We call this side-by-side model simply because, the way we make it kind of, my student was very ingenious, like he uses tilted surfaces so that he can put the spheroids at an interface, but then he can image them in the XY plane. Because if you've done a microscopy imaging in the Z plane and analyze that, it's not very easy. That's actually when you have like 3D models, microscopy is like your one way to look at data, and if you can't do microscopy very well, like you don't know what to do with your data, right? So that's actually, the XY worked well. We put the spheroids inside. I won't go into these materials, like we had soft, we had stiff materials, soft like normal brain tissue, stiff like glioblastoma. We put spheroids in there. The spheroids were doing fine, like very good cell viability. They could spread. This is just a little bit of blow up so that you can see the spheroids. You put spheroids both in the soft and the stiff gel just to see what they're going to do. And you can see that they can cross the interface, but also if you kind of look at the cells, some of them are just going along that interface. And something which I'm not going to talk about today, we have a whole other story on that, but the cells that are migrating, they become cancer stem-like. So not only they're migrating, they become resistant to chemotherapy and treatment. Anyway, a little bit more on like crossing the interface. Cells seem to cross into the stiff, but not into the soft. And when we did drug screening, I will only point your eyes towards the very last graph. If you simply look at global, like spheroids, there are cells in the core, cells in the periphery, the interface, in the middle. So if you look globally, like all cells respond the same. But if you now look specifically at the core versus the periphery, we saw something really interesting. They responded differently. And so if you know anything about cancer, like the rule of thumb, right, even if you have breast cancer or something, it's like the easiest, you know that you have cancer because you can feel it, right? Like the cancer makes the matrix stiffer, because cells remodel the matrix. In fact, even the cells even produce collagen in your brain, which you don't usually have. You don't associate collagen in your brain. So the cells make the matrix stiffer. So the hypothesis was that those migrating cells that go into that stiffer environment will also be more resistant to treatment. And I'll just have it here spelled out for you so that you can read it. What we noted from our data is that, yes, matrix stiffness does matter. And we specifically looked at invading U87 cells, but basically in the stiff, we saw that invading cells, basically, here. So invading U87 cells, which are the ones that are migrating from the core into the interface in stiff, are going to have drug resistance, but not in the soft. And the stiff is just like, kind of like, like in vivo. Anyway. Let me see. I have 3 minutes. I don't know. Let me see. I don't have 3 minutes. I'm just going to skip this. So this was a whole other story just to tell you. And you just have to believe me on this. So I'll just give you the very, very last slide right here, is that something to keep in mind. If you're putting cells in materials, you have to think about time. And this is something that we don't usually think about. So we did a whole study basically looking at different times, like how many times you need to passage the cells in a specific environment to let them adapt. And the way I think about this is, if I give you a new job and I throw you into that new role and ask you to perform, and I judge you on day 1, you might not do as good of a job. But if I judge you on day 9, like in this case, you probably are going to be more accustomed to your new role and you're going to be doing better, right? So the same is with the cells. And here I just wanted to show you briefly, those are breast cancer cells, by the way, and those were cells which were preconditioned to the environment. So if you simply let the cells adapt to the new environment, then they will behave much differently. And it's all a mechanosensing field, but you probably have heard that if you put cells in a soft environment, they stay around, and if you put them in a stiff, they spread. Kind of like if you have heard about this overall rule of thumb. And this is simply because of the contrast force that cells can exert on their environment. Well, it's not the case. If you precondition the cells to the soft environment, they spread just as well on it. You simply have to condition them to it. And anyway, so this is going to be my last slide. And just thanks to my lab, like everybody who is actually doing the work while I'm writing the grants to fund the work, right? And Also, our collaborators, specifically for this work, we have several which are listed right here, and all of our funding. And with this, if I have time for questions, I'm not sure, but thank you.
Okay, got time for a question or two? I know we're headed off to a business meeting soon.
Thank you for this great presentation. It was very informative. I have a question about using this hydrogel for delivering RNA therapeutics. Are they better alternative also?
So RNA and DNA delivery is just a little bit different because our hydrogels, they're more for localized, they're micro size or bulk materials, so they're more for localized delivery. They don't enter the cell. And so you can position your therapeutic in the proximity of your disease, like what you're targeting, but they don't actually enter the cell. We have to make them much smaller. We actually can use ATPS, which is like a different aqueous to phase separation, to make 2 micrometer microspheres, and those could be endocytosed, but we haven't done anything with RNA. So really the limitation is like, can they be endocytosed or not? It's the size.
Thank you.
Your homemade rheometer, the ball-bearing rheometer, so I was kind of interested in that. So for your bottom compartment that you 3D printed and you put the 3 ball bearings in there, do the 3 ball bearings spin, or do they just kind of like rotate at all?
No, they are stationary.
They're completely stationary? They don't move? Because the top one you got anchored with some adhesive tape, right? And then.
Right, so all of those are stationary, and so the top one just rotates, and basically, so the whole thing works because you are looking at lubricious liquids or like, salt gel formulations, slurries. So anything which is liquid, like you have to fill the balls. And so it's really, there's nothing rotating. It's simply
those
the
ball
diameter.
bearings on the bottom don't. I was wondering if that was spinning and how you're calculating then your
No, it's
your
not spinning
friction
at all. Yeah,
coefficient.
we have a lot of data showing, we basically have to change them after every measurement because we don't want any scarring that changes the measurements. Yeah.
Great talk. Really quick question, this might be very simple. You're saying that these glioblastoma cells, can clearly tell the difference between stiff and soft. Is that all cell types? Is it specific to the glioblastoma?
Not all cells. Cells in general respond to stiffness. That has at least most cells, not all. But specific to glioblastoma is that they migrate along those interfaces. So yes, it's specific to glioblastoma.
So do you think it's something on the surface of the cells? Like, do you have a hypothesis of what?
There are multiple hypotheses. So some people also it's also biochemical composition is a little bit different on those interfaces. It's not just the stiffness. I don't want to speak to that now, but we can talk separately. There is some stuff like CD44 is important, for example. But there is not conclusive evidence why that is.
It's a nice talk. I'm curious about the remodeling constantly going in the extracellular matrix, and especially in any kind of pathology, whether it's inflammation or is it a cancer or infectious disease, how these hydrogels could be useful. One of the things which I was thinking could be blind is that one of the enzyme called heparinase, which is involved in digesting heparin sulfate and causing jungles of these heparin sulfate in the extracellular matrix. But they have been implicated in cancer pathology. So I'm curious that your hydrogel could be used as a drug, as anti-inflammatory gels to prevent inflammation.
Are you thinking in the context of a drug delivery, or in the context actually as a matrix, like cellular?
So
cellular
myself
matrix.
will be very much struggling to say it could be localized, it could be internalized, but because heparinase get activated during the inflammation, I was thinking that hydrogels could be encapsulated with this anti-heparinase.
Oh, yes. Okay. No, I should talk about this with you more after, because it's very interesting. Yes, absolutely. So any large molecule would be a great candidate for those materials because, again, those are nanoporous materials, the nanostructure, they're about 5 to 10 nanometers in diameter. So you can release sustainedly any kind of large molecule, and that could be controlled.
The reason I'm saying is because many viral infections now, apart from cancer, they have shown that these cells or tissues, they have high level of haprenase, tissue remodeling, and as a
Yes.
result of, you have a lot of inflammatory cells at that localization.
No, thank you so much. I'll talk to you later.
Okay, let's thank our speakers again. Thank you.